Talk about the plant growth hormones. In the plant growth hormones, we otherwise call it as plant growth regulators, phytohormones. So these are the diversified chemicals. The list of the phytohormones are, we have two types in this: plant growth promoters, plant growth inhibitors. So coming to this two, in the plant growth promoters we have auxin, gibberellic acid and cytokinin. Plant growth inhibitors are abscisic acid, ethylene. So coming to the derived compounds, from which compounds these are derived? Auxin is derived from indole compounds. Gibberellic acid is derived from terpenes. Cytokinin is derived from purines, which is adenine. And abscisic acid is derived from carotenoids. And ethylene is derived from, it is gaseous, volatile. So let's talk about the very first one, auxin. So coming to the auxin, this is an indole compound. Remember the four compounds: IAA, indole 3 acetic acid. And next IBA, indole butyric acid. So these two are naturally occurring. And next NAA, naphthalene acetic acid, and 2,4-D, 2,4-dichloro phenoxyacetic acid. So these two are synthetic. So coming to the physiological effects of auxin. So physiological effects of auxins are: initiates roots in the stem cuttings. So remember how to remember these auxins are. Remember from the root, stem, flower, fruit, seed, like that. So then you can remember this. So roots in stem cuttings. And roots will be absorbing water and minerals, and that will send through xylem. So cell division. So the cell is divided, finally differentiated as xylem. So xylem differentiation. Afterwards stem. For stem, remember apical dominance. Coming to the application, apical dominance. So removing the apical bud. In tree plantations, for hedge formation. This is application for removal of apical bud. Next, afterwards, remember about the flower. Initiating flowering in pineapple. For fruits. So flowering is done for fruits. Parthenocarpic. Tomatoes. For seeds. For conifers, hastening the maturity and speeding the maturity. For seed formation. And finally, if you come out of the plant, this will be acting as a weedicide for dicots and do not harm matured monocots. So remember one thing, the discovery of this. Sorry for making it in a final note. Auxins are first extracted from human urine, first observed by Charles Darwin and his son Francis Darwin in coleoptiles of canary grass. And also F.W. Went conducted experiments on coleoptiles of Avena, which means oats, Avena sativa, oats. So we call that test as Avena curvature test. So next it is gibberellic acid. For this, there are nearly 100 compounds of gibberellic acid and they are named it as GA1, GA2, GA3, etc. etc. But GA3 was first structurally characterized. And it is first discovered by, so discovery, Kurosawa So it is first discovered by Kurosawa as Bakane seedlings disease or foolish seedlings disease in rice plants. So which is caused by the fungus Gibberella fusicuroi, and he infected the normal rice seedlings and observed the symptoms. So let's see the physiological effects. For gibberellic acid, I'll tell you an easy mnemonic. Remember G and A. G for grapes. So excuse me. There is one point. Actually, this point I have given something wrong. Excuse me for this. This is for gibberellic acid. For cytokinin, the point is flowers, fruits, and leaves. In case of immature, it will stay on the plant. In case of maturity, it will promote abscission. So that is the thing. Excuse me for that. And A for apple. Remember grapes. Stalk length is increased. Sugarcane, internodal length is increased. And here one more C for conifers. And for rosette plants such as beetroot and radish, excuse me, beetroot and cabbage, internodal length is increased. And for apple, elongation and fruit shape setting is promoted. And for conifers, hastens the maturity and forms seeds. And gibberellic acid is antagonistic to abscisic acid. So they both are antagonistic to one another. So this is gibberellic acid. Thank you.